In Activity 12, Death of Organisms, students observe that even dead organisms have a valuable role in the cycles of life. Students first predict what will happen as organisms decay and then observe the decomposition of dead organisms. Finally, students infer that in nature, the decomposition of dead organisms enriches the soil. You will need the following materials from the kit. Activity Sheet 12, Parts A and B, 8-ounce paper cups, magnifiers, petri dishes, tweezers, disposable kit bags, masking tape, transparent tape, and water sprinklers. You will also need to provide newspaper, samples of food, local soil, and tap water. To prepare for Session 1, make a copy of Activity Sheet 12, Part A for each student. Fill the water sprinklers with tap water. Gather and bring to class samples of food that will readily decompose, such as bread, cheese, and vegetable peelings, in sealed plastic bags. Choose a warm location where all petri dishes may rest undisturbed for approximately 4 days after Session 1. To begin Session 1, review with students the stages of a life cycle. Then ask, during your study of fruit flies, what observations did you make about death? Students should have observed that not all eggs hatched into larvae, not all larvae became pupae, not all pupae matured into adult flies, and some of the adult flies died without producing a new generation of offspring. Then ask, during your study of pea plants and corn seeds, what observations did you make about death? Students should recall that not all the pea seeds and corn seeds sprouted, and that some of the plants died during the course of their life cycles. Display the food scraps and ask, are these living, non-living, or dead objects? If necessary, point out that the food scraps are parts of or made from things that were once alive. Therefore, they are dead things. Divide the class into teams of 2 and give each team a petri dish, a strip of masking tape, and an assortment of food scraps. Instruct students to put food scraps into their petri dishes and label their petri dishes. Have students place their open petri dishes with the lids next to them for identification purposes in an undisturbed place for approximately 30 minutes. This time will allow bacteria to settle on the food scraps, speeding up decomposition. Then give each student a copy of Activity Sheet 12, Part A, and instruct students to work independently to complete it. When students have finished, allow time for them to share their predictions. After 30 minutes, direct the class's attention to the petri dishes. Distribute the water sprinklers and tell students to moisten their food scraps. Students should then close the petri dishes and secure the lids in place by encircling them with strips of the tape. Have the class place the dishes in a warm, dark place, such as a closet, and explain that in a few days they will check on the predictions they have made. To prepare for Session 2, make a copy of Activity Sheet 12, Part B for each student. Fill a paper cup with soil for each team of 2. Gather enough samples of fresh local soil containing pebbles, live organisms such as ants, earthworms, and decaying organic matter. Make sure to begin this session once a significant amount of mold has formed on the food scraps. To begin Session 2, have each team retrieve their petri dishes and a magnifier. Make sure the students do not open the dishes. Instruct the class to examine their food scraps and ask, what changes do you observe? Students should notice a white, pink, or blue fuzzy or powdery substance growing on the food scraps. Encourage students to observe other petri dishes, noting how different foods may have undergone different changes. Inform students that the food is beginning to decay or break down, and explain that bacteria are tiny organisms that help to break down or decay dead organisms. Ask students, do you see any mold in your Petri dishes? Encourage students to identify the fuzzy or powdery growth on the food scraps as mold, and explain that mold is a tiny organism that feeds off of other living and dead organisms. Explain that during the 30-minute period that the food in the Petri dishes was exposed to the air, bacteria and mold particles in the air settled on the food. Distribute to each student a copy of Activity Sheet 12, Part B, and instruct students to complete Question 3, including the drawing and the description of the changes they observed. Ask students what would have happened to food scraps left in a natural setting. Students should predict that the food scraps would have probably begun to decay. Ask students, what do you think would happen to food scraps if they were buried in the soil? At this point, accept all reasonable responses. Students may speculate that the food scraps would continue to decay until they became tiny pieces of the soil itself. Then, tell teams to return their Petri dishes to the closet and then to cover their desks with newspaper. Distribute to each team a pair of tweezers and a paper cup filled with the local soil. Have students dump their soil sample onto the newspaper and examine it with their tweezers. Ask students what is contained in your sample of soil. Students should be able to observe twigs, pebbles, bits of dead leaves, and perhaps worms and insects. As students report on their observations, work with them to classify each object as living, non-living, dead. Point out that the non-living substances, various types of rocks and minerals, are basic elements in the Earth's surface. Further explain that living organisms, such as ants, beetles, and worms, burrow into the soil and also live there. Lead students to understand that some of these things eat other living organisms, and others eat dead and decaying plants and animals. Bring the class's attention to the fragments of leaves in the soil that have broken into very small pieces. Ask students, why do you think this happens? How did the large pieces become small pieces? Students should speculate that the leaves decay, breaking down into tiny fragments that become part of the soil itself. The same thing would happen to pieces of fruit or vegetables that were buried in the soil. Ask students, how might the decaying parts of dead plants help new plants to grow? Help students realize that many dead organisms contain nutrients or helpful materials that living things need to grow and survive. When the tiny particles of decayed plants and animals mix with the soil and the water that is in the soil, the nutrients feed the new plants and help them to grow. Based on their observations, instruct students to complete questions 4 and 5 on Activity Sheet 12, Part B, and then discuss their answers as a class. To conclude Session 2, students should dispose of their dirt samples, then have them wash their hands and return the paper cups, magnifiers, transparent tape, masking tape, and tweezers to the kit. Collect all Petri dishes in one of the disposable bags and seal the bag and attach a contaminated material tag to it. Use the other disposable bag for the fruit flies. Finally, place all the vials in the bag and then seal the bag and attach a contaminated material tag. Make sure to freeze the contents of the bag before disposing of it. For science background, reinforcement activities, curriculum connections, and information about the Delta Science Reader, please consult your DSM Teacher's Guide.